Okay, so we did all kinds of passive transport, which is over here on the left-hand side of this picture, right? And we did all kinds of active transport over here on the right, right? So, and then remember, this is just an imaginary line, and you can spell that any way you want. But that imaginary line is just saying passive over here, active over here. We did talk about primary active transport, which is against the gradient. And that is the biggest difference between active and passive transport. Passive transport is with the gradient. And we learned that primary active transport is about moving one molecule up its gradient, against its gradient, in the direction that it would not flow with no energy. Now, the thing is, primary active transportation, you could call it carrier-mediated, yup, but all active transport is carrier-mediated. And the other thing to know is that just like the shape that changes in the facilitated diffusion, this protein here also changes shape. However, that shape change incorporates the use of ATP to happen. This changing shape over here in facilitated diffusion is kind of like a trapdoor, right? You step on it and it opens. So water goes up to it and it opens. But over here, these molecules that we're pumping into the ICF, we need a shape change to move the molecule, but we also need ATP, and that's what makes it active transport. So in secondary active transport, there's a molecule that we're meaning to move, but there's another molecule that kind of comes along with it. What I'm going to draw here is I'm going to draw a whole bunch of molecules outside of the cell, and not too many of them inside of the cell. And then I'm going to draw some other molecules. And there's going to be tons of them outside of the cell, and not too many of them inside of the cell. Now the problem is, this protein here is actually geared to recognize just one of those molecules. And so one of those molecules, like glucose, it links on or docks itself at the protein, and the protein sucks up some ATP and pumps that molecule into the ICF. Now, just like a hitchhiker, like a real one, like a burdock that gets on your pants when you go through the woods, this other molecule here, which we could say is salt, this other molecule just kind of like hitches a ride and goes with it. And this is actually what happens with salt and glucose in the kidney. And this, right here, this little protein, is the source of many issues with a diabetic, but it is also the target of many drugs like Invokana. Okay, so this is what is called co-transport. So this, let's give it the full name. This is carrier-mediated secondary active co-transport. That's what this is. There's also something that's called anti-transport. And this is where we are also moving molecules against their concentration gradient. So let's draw a whole bunch of the green molecules in here, and not so much of them out here. And let's do the opposite. Let's do just a few purples in here and a ton of them out here. And so what we're going to do is we're going to use ATP. So again, let's note ATP because we're talking about active transport. But we're going to use ATP, and we're going to use it to pump the green molecule against its concentration gradient into the cell. We're also going to use ATP to pump this purple molecule against its concentration gradient outside the cell. And that is what is called carrier-mediated secondary active anti-transport. Now, I'm going to stop this video, and what I'm going to do is make another short video on the sodium-potassium pump.